In this webinar, you're going to be hearing from Dr. Miranda Steele and Dr. Sander Wuyts about their work decoding TCR specificity using predictive machine learning models. A little bit of background: Miranda leads operations and R&D at iRepertoire, co-developing key technologies like iPair and RepSeq+. She holds a PhD focused on protein biochemistry and has authored patents in single-cell analysis, antigen-specific antibody detection, and multiplex amplification. Sanders is CEO and co-founder of ImmuneWatch, an immunoinformatics company specializing in T-cell receptor repertoire analysis. Their software, ImmuneWatch DETECT, is the leading solution for T-cell receptor specificity annotations, accelerating immunotherapy development. I will go ahead and pass it over to our first speaker, Miranda.
Thank you, Jenna. Thank you, everyone, for attending today. I'm very excited to share some of the work that iRepertoire and ImmuneWatch have done together. So this is going to be broken up into two parts. On the first part, I'm going to talk about the various kind of wet lab methods that we utilize here at iRepertoire to generate the TCR repertoire data, and also single-cell specific iPair data. And then Sanders is going to go into the computational approaches to actually mapping those antigen-specific TCRs. So for those of you who don't know us, iRepertoire is located at the Hudson Alpha Institute for Biotechnology in Huntsville, Alabama. We are all about the immune system and immune profiling, and we have several technologies related to that, which I'll discuss today. We are an end-to-end facility, meaning that we will perform services all the way from the initial sample through the final data analysis. We do this through both through our services of bulk repertoire and our single-cell VDJ analysis, along with standard data analysis that we provide in addition to exploratory analysis packages. We have three primary offerings. I'm going to talk mostly about our bulk repertoire, or the majority of the approaches that we're talking about today are focused on bulk repertoire, and this is where we have bulk RNA, where we're examining the VDJ sequences. Unlike other platforms that are on the market, we have a massively parallel approach to this so that we can cover both T-cell repertoire and B-cell repertoire all under the same amplification conditions. So you get the TCR alpha, beta, delta, gamma, Ig heavy, and kappa lambda, all under those conditions. And then you can go on and apply various computational approaches to that data. Our single cell approach is called iPair. This is usually coupled to flow cytometry, where we're picking out these very rare antigen-specific T cells or B cells and depositing them as single cells on a plate and co-amplifying the receptor pair. And our final platform is called Immunocyte. This is a targeted RNA-seq panel that covers over 150 targeted genes for phenotyping expression. This can be applied to both bulk RNA or to our single cell platform. So why is detecting antigen-specific TCRs important? I think everybody here knows this, but I think it's good to review some of the reasons why we should do this. When we're trying to map immune responses, picking out those TCRs that are actually driving the protection or the pathology of therapy response is very useful in terms of their dynamics. One, knowing what they are, and then determining how their trajectory changes throughout time. This actually enables the second goal, which is biomarker discovery. Instead of just tracking an overall broad changes in repertoire, which is useful, now you can actually track the exposure, disease progression, and treatment efficacy with high precision by looking at these particular markers. If we go to the more specific approaches, such as the single cell approaches, these provide the starting points for TCR engineered therapies, vaccines, in immuno-oncology as well. And then finally, this leads to advanced precision medicine, where we can start linking patient-specific TCR outcomes to tailored interventions. And that's, I think, much further downstream, but it's still definitely in sight. So why has this problem been so difficult, and has it actually been solved? So the number of T cells that are discovered in a given repertoire experiment is very, very large. So you can get a list of upwards of 100,000 unique clonotypes. The problem is the TCR is only one side of the equation. So how do you map each of those TCRs to the epitopes that they are recognizing is a very, very difficult problem in immunology. There's also other challenges. There's crosstalk among TCRs. there are likely multiple TCRs that are capable of responding to a given epitope. So you're trying to deconvolute that information. And has this problem been solved? I would say no, but I'd say we're definitely further down the path to solving it. And we've identified various tools to address these questions. These methods are constantly evolving and it remains a challenging problem. But once you have your repertoire data, it's something that you can interrogate repeatedly as databases improve over time, which I think is a really important point. So I mentioned in the beginning, we have a massively multiplex approach to this. We cover both T cell chains, alpha, beta, delta, gamma, Ig heavy, kappa, lambda. We try to capture the full length VDJ receptor. On B cell, we also capture the isotype, which is a really important piece of information. So that's just showing our coverage on our primer sets. And as I mentioned before, we're not just getting a picture of the beta or the alpha only. We're actually getting a full kind of perspective or a holistic perspective of the immune repertoire where we're covering all of the B cell chains, Ig heavy, kappa, lambda, TCR alpha, beta, delta, and gamma. Our single cell approach is flow cytometry based, where we're using flow cytometry to pick out these very rare cells. So they could be as infrequent as 1 in 10,000 cells, depositing them on the plate as single cells and recovering the paired VDJ receptor sequencing. So this is going to the much more specific where you're actually maybe trying to develop a drug and you really, really want to pin down the identity of a rare cell population that might be clonally expanded against that particular epitope. So I like to go over a slide that I like in terms of how I think about antigen-specific solutions and some of the conversations Sandra and I have had with regards to this. So if we think about the approaches in terms of specificity, if we're looking at this kind of bullseye, on the outer circle, we're talking about things that maybe are less specific. This would be repertoire data with no training data. And you could be utilizing temporal changes of the immune repertoire to look at emerging T cell and B cell clones. You could be applying other models that maybe aren't using training data, such as Sonia Olga or DETECT using motif mode to identify putative antigen-specific clones, but you haven't really applied any sort of training model to that. As we go a step in, this would be taking repertoire data, so no single cell data in any of these approaches these first three approaches. You're taking repertoire data in conjunction with epitope TCR prediction models, such as ImmuneWatch DETECT that you're going to hear about today, and publicly available epitope TCR databases, and using those then to predict antigen-specific clones. But it really starts to get much more specific as you start to train the data sets with your specific epitopes of interest. And to do this, you can basically stimulate PBMC samples with various epitopes and look at the emerging clones and then use those as the training data set for the ImmuneWatch DETECT platform. And in that approach, you basically are becoming more and more specific and better accuracy in terms of determining what are the ag-specific clones that are within your data. If we go even further into this pool, we start to get into more wet lab approaches that involve some sort of sorting up front. So I could be sorting antigen-specific clones using a pool of antigens, but I'm actually pulling out those cells and maybe sequencing them in bulk. I'm not worried about the paired receptor. I can definitely use that as training data. It's going to be very specific. And then I can go really, really deep into this where I actually go to the more drug discovery approach where I'm actually pulling out these antigen-specific T cells or B cells and basically uncovering their paired receptor sequence. So at this point, I'd like to hand it over to Sander, who is the best authority in terms of discussing the DETECT platform. So Sander, I'm going to stop my share and let you take over.
Thank you, Miranda. That was great. Excited to be here. Thank you so much for the invitation as well. So for the next part of the presentation, I will tackle two topics. First of all, I will look into how these models work. And secondly, I will also present some kind of use cases where we effectively applied these T-cell receptor specificity models. But first of all, to get to know the audience a bit better, I'd like to run a quick poll. Have you ever used TCR epitope prediction models before? I will come back to that question later on in the presentation. Okay, let's dive into the first topics. Let's start with the models and the algorithms. To frame this a bit better and to absolutely make clear where this kind of machine learning models or TCR specificity models come into place, it's always at the end of the T-cell receptor sequencing process. So usually you start with your T cells. This can come from bulk PBMCs or from TILs, for example. You do your sequencing and you end up with what we call an unannotated T cell repertoire. It's only when you actually constructed your T cell repertoire by assembling all of your different reads into clonotypes, a tool like ImmuneWatch DETECT can come in. We basically take this unannotated T cell repertoire as an input and then output an annotated T cell repertoire where we try to fish out viral specific TCRs or, for example, tumor specific TCRs. Even more in practice, it looks like this. So the input is usually your clonotype file, which is a collection of your CDR3 sequences. We call them junction AA here, a V gene call, a J gene call. And what we then add with a tool like ImmuneWatch DETECT is we'll add the epitopes that we believe that this specific TCR or clonotype might bind to, a score, the antigen that is related, where this epitope has been derived from, the species, the HLA type that this epitope is usually presented in the HLA context, but then lastly also a reference to this is a pointer in our database that shows that this specific TCR has been described in this paper belonging to this epitope. So it gives you a little bit more explanation on how we came to this match instead of just being a black box model. Now, these TCR epitope algorithms, they come in different kind of flavors with also varying performance. And I try to classify this kind of algorithms into using two different axes. We can classify them according to their speed, so from slow to fast, but also whether they are performant on the seen epitope use case or the unseen epitope use case, shown here on the x-axis. Speed should be obvious what we mean with this. Some algorithms, like for example, AlphaFold 3, will take like a half an hour to run one single TCR on the most fastest GPUs available, while a tool like ImmuneWatch DETECT can actually annotate 100,000 TCRs in just under a minute. On the x-axis, it might be a little bit more difficult to just understand out of the box here. So I'll try to run you through this. We're talking about how performant an algorithm is in the unseen epitope condition. Seen epitopes are typically models that can do predictions only for epitopes stored in their database. You need the right training data to be able to do these kind of predictions. For example, famous antigens such as the spike protein for SARS-CoV-2, or well-characterized tumor-associated antigens such as MORD1 or NY-ESO1. Usually, we have some example TCRs that are known to bind these epitopes, and from this you can train a machine learning algorithm, which is usually called a seen epitope prediction algorithm. Most of the algorithms in this space can be found here, like Glyph2 together with VDJDB, but there's like many more algorithms that fit into this space. The holy grail in system immunology is also this unseen epitope predictions, but as you can see, this space is rather blank because we still believe this is a very unsolved problem. ImmuneWatch DETECT has seen some performance in this case, but there's also these structure models like AlphaFold 3, Chai1, Boltz1. They have been showing some relatively okay performance in this space, but nevertheless, it seems to be an unsolved problem. How do we know this? Some experts came together and actually organized public benchmarks to see how these kind of algorithms actually perform. Because we see a new type of algorithms being published every 3 months or so, and it was really very hard to evaluate which ones did perform well and which ones not. So that's why a group of scientists came together and started organizing these IMREP challenges to independently assess the performance of these type of algorithms. And we've seen 2 major benchmarks in the last couple of years, IMREP 23, which was organized for a seen epitope prediction performance, and IMREP 25, which was organized to also evaluate the unseen epitope performances. These are the results of the IMRAP 23 benchmark, which is also being published the end of last year in a journal called Immunoinformatics. So if you want to learn more, feel free to read that paper. And these are not my words, but this is literally what's been put down in the paper. ImmuneWatch DETECT shows a substantial predictive advantage compared to all other models evaluated in this competition. And one of the main conclusions is that these seen TCR epitope models actually achieve relatively good performance and work very well on the condition that you already have some kind of training data for an epitope of interest. And that ImmuneWatch DETECT is best in class in this type of algorithms. Now, if you switch to the IMRAP 25 competition, the best performing algorithms are structural models, AlphaFold 3-like models. You can see here, Chai-1 is also one of these, or for example, we use the Bolt-1 combination. But in general, the conclusion of this competition was that unseen TCR epitope performance is very, very poor in general. And that even though these structural models perform very well, they're also very impractical to just annotate the complete repertoire. And so the conclusion is currently for now that it's still an unsolved computational problem. And the manuscript of this competition is still under preparation. That's what I heard last of it. So it might be appearing in the beginning of next year. Now, coming back to this IMRAP 23 challenge, why is ImmuneWatch DETECT so performant? I think there's two reasons, but the main reason is quality data is key. We founded ImmuneWatch 4 years ago, and the initial focus was generating our own in-house database of epitope TCR interactions. For this, we gathered more than 2,100 different kinds of epitopes. 50%, for example, are coming from viral sources, but 50% is also non-viral sources. I already mentioned TAs like NY-ESO1, MART1 is in there. And recently, we've been focusing a lot on adding KRAS-specific epitopes. And this led to a database of more than 200,000 TCRs, which we curate ourselves from public datasets. And we try to release a new set each quarter, with the latest release being just last week. An extra thing we're doing now is also we're generating our own in vitro data ourselves, and we hope to expand that in the future as well. But the goal is to keep the database as up to date as possible. And so we're already looking at a backlog and trying to get every paper that has T-cell epitope data or qualitative T-cell epitope data in the database. And for this, we use a modern annotation pipeline, basically by executing a search every month on PubMed, trying to find the relevant papers out there, classify them using LLMs to actually extract the right information so that our manual curation team can efficiently curate them and see whether these data should be added, yes or no. Now, a database itself is not enough. You also need an algorithm to be able to query this database. And this is where our expertise, our academic expertise comes in, because we've been working on this problem for more than 10 years now before spinning out in a company, ImmuneWatch. And we've been developing this kind of algorithms already for a while now. And the central concept is actually quite simple. If you think about it, it's similar TCRs bind similar epitopes. So it all starts with our database that I just explained. But in the second step, what we do is then if you take a query TCR, for example, here defined as a TRB-V gene, so a V gene and a CDR3 sequence, we then usually cluster this together with our database, which is what you can see here. And then in this local space, apply a new machine learning algorithm with some scores coming from our database to actually get a confidence score on how confident we are that this specific TCR might bind to a certain epitope found in our database. So this should give you a little bit of an idea what's under the hood. Okay, that was the machine learning models. I'd like to switch now to several use cases, 3 use cases to be more specific on this, on how we apply these kind of models. But first, I want to have a quick check at the poll to see how many of you have already used T-cell epitope prediction models. And I see that around 70% have not used them, but some 30% have used similar models before. That's interesting to note. Okay, use cases. I will be presenting 3 use cases, and as Miranda mentioned, I like this kind of way of showing things. I left out the inner circles because these are very wet lab intensive protocols. They're super valuable to create antigen-specific repertoire data. But now we're focusing on what can we do in these outer layers using computational models only. I've brought 2 use cases here where we basically just use our database in IMWDB, and 1 use case where some data has been generated in-house. During the presentation, I will briefly also show 1 example of where we did not have any kind of training data, but I'll point to that later on in the presentation. So use case 1 is, I have 2 blocks. 2 use cases will be on immunomonitoring, one of the most frequent use cases that we see ImmuneWatch DETECT being applied to. And a 3rd use case will be on TIL product quality checks. Why do I like presenting immunomonitoring cases? Because I firmly believe that TCR sequencing is rising to begin the gold standard in immunomonitoring, which has also been shown in this hallmark paper published in 2023, where the authors, some of them belonging to BioNTech, compared T-cell sequencing on the top row to ELISpot on the bottom row and found it to correlate it relatively well. So T-cell sequencing data actually correlates relatively well with ELISpot wet lab data. But T-cell sequencing will give you more information, requires less hands-on time, and it's probably more sensitive to analyze the T-cell response compared to a tool like ELISpot. Now, how can you use T-cell sequencing for immune response monitoring? Well, as Miranda has shown, each clonotype can actually be typed by a unique T-cell receptor. This can be an alpha chain, a beta chain, a gamma chain, or a delta chain, or a combination of these chains. And if you look at a certain event, for example, a vaccine or cancer therapy, then you might see some shifts in the frequency of clonotypes. For example, this blue clonotype goes up in frequency after a vaccination campaign, then this might be a relevant clonotype for the T-cell, for studying the efficacy of your therapy. So the first use case will be on a SARS-CoV-2 vaccine that we did in collaboration with iRepertoire. Basically, they ran this COVID vaccine study where they took samples at baseline, did a first shot, and then had 4 different samples for which they submitted to T-cell sequencing after the vaccine shots, then had a second dose with, again, 4 different time points, 3 days, 7 days, and 14 days after the second dose. Again, showing you the process a little bit. You start with the T cells, you do T-cell sequencing, you get your unannotated repertoire, and then used ImmuneWatch DETECT to try and annotate the SARS-CoV-2 specific clonotypes. So this is the work done at iRepertoire, and only the last step is applying ImmuneWatch DETECT on the data. This is a bit, this is what the data looks like in practice if you really want to dive in. So these first 3 columns are the basic clonotype files that you would get out of a typical T-cell sequencing experiment, with the first column showing the CDR2 sequences. We have the V gene, the J gene, and then the frequency of each of these clonotypes. And then this is the new information that we add with ImmuneWatch DETECT, an epitope that is most likely to be the binding epitope of this specific TCR, a score on how likely we believe that this epitope binds this TCR, the reference TCRs, and the antigen and species where this specific epitope is being derived from. Now, in the context of this specific vaccine study, we were interested in only looking at the SARS-CoV-2 specific clonotypes. So we can easily apply a filter. You can even do this in Excel if you want to, and then filter on species equals SARS-CoV-2. And now if you take all of these clonotypes and then plot the frequency in time for each participant, you actually get a plot like this. For example, each panel is one different patient or participant. And let's have a look at this bottom left panel. participant 14, where you can see that there's almost no SARS-CoV-2 specific clonotypes at baseline, but you see that the frequency of these SARS-CoV-2 clonotypes will increase 14 days after the first shot, decreases again, and then increases suddenly 7 days after the second dose. And this is completely in line with what one would expect of infectious disease vaccine, where you get some, where it takes up to 14 days to build immunity after the first shot, but the second wave of immunity comes much, much faster as the T cell response has been already initiated after the first dose. Now, an interesting side effect here, we were looking at SARS-CoV-2 specific vaccines only, but because ImmuneWatch DETECT has a strong database behind it, we also noticed that in patient number 3, there was this sudden increase of EBV specific clonotypes. And we were not screening for these specific clonotypes, but because of this big increase, we told the iRepertoire team, hey, we're seeing some EBV specific clonotypes there. And they later confirmed us that this person indeed suffered from mononucleosis, which is an EBV-induced disease, showing that we can do a lot more than just looking at specifically SARS-CoV-2 specific clonotypes. So that's it for use case 1. I've brought a second use case more in the oncology field, which is more in the inner ring, because in this use case, we are looking at a neoantigen vaccine where training data was generated to retrain ImmuneWatch DETECT so that we could also track the epitopes specific to this neoantigen vaccine. And this use case was in collaboration with the Centrum for Human Genetics in Tübingen in Germany. And basically, they created a personalized cancer vaccine composed of different specific different cancer-specific epitopes based on tumor mutations. And they found that this vaccine actually had a very good clinical response. Okay. Like iRepertoire, they also took samples pre and post vaccination. So they had one sample pre-vaccination and a couple of samples after the personalized cancer vaccine was given to this patient. T-cell sequencing was done on both blood samples and tumor samples, and a subset of PBMCs were used for epitope stimulation. So they really went into the wet lab and stimulated these PBMCs with their epitopes of interest, then submitted these samples to T-cell sequencing, give this data to ImmuneWatch. We retrained the model so that we were able to track the T-cell response specifically to those epitopes used in their cancer vaccine. And here we're focusing on just 4 different epitopes in the vaccine, peptide 19, 17, 20, and 21. And we see a clear increase in frequency of clone types mapping to peptide 19 and 21, especially in the post-2 samples. And we didn't see any kind of increase for the peptide 17 and peptide 20. Showing that that we believe that especially peptide 19 and 21 were causing the T-cell response, which was also later confirmed in follow-up in vitro experiments. I have plenty of more use cases on immunomonitoring, so please go to our documentation pages if you want to follow along with these kind of tutorials. And I just want to highlight one of them, where we also have a neoantigen vaccine without data, which is more in this unseen epitope prediction space, which is a very, very hard field. But we've seen some interesting results there, so feel free to check it out. Although I want to stress that this is usually a much, much harder problem compared to the use cases that we've seen here. So that brings me to the last use case that I want to present here. And this is not about immunomonitoring, but about a TIL product quality control, where we've brought a use case on melanoma. And I quickly want to give an overview of what a TIL therapy exactly is. Basically, people that administer TIL therapies, they take some tumor samples, a biopsy, and they do in the lab, they will do in vitro expansion to generate more T cells coming from the tumor, and then we'll try and re-infuse these patient cells as a sort of therapy to try and kill off the tumor. So the goal is to stimulate tumor-specific T cells in vitro. Now, People in the TIL fields, they like to use T-cell sequencing, and they use it for two different use cases. The first one would be product characterization, and the second one would be immune response monitoring. The product characterization is interesting because you can then have a look at the clonality and diversity of your in vitro expanded TCRs. You could also annotate the specificity because this would give you some insight into the product quality, but can also help you identify relevant clonotypes. You could then also use this information to actually track this TIL expanded of in vitro expanded TILs in the blood of the patients. But since I've already shown two use cases on immune response monitoring, I will just stick to product characterization in the next coming slides. For this use case, we got some public data from this paper from the Kukos and Harari lab in Switzerland, where they applied a TIL therapy for a melanoma patient. We've obtained 4 different samples. 2 of them come from the TIL product, and 2 of them were from the pMHCs, but I'm going to skip that in this presentation. And if you look just at the T-cell product, I think that's this specific sample. You can see that the first thing we usually do is cluster all of these clonotypes into different clusters. And what's already interesting is that these couple of clusters, they already take up 70% of the whole T-cell repertoire, showing that these are very, very evident clones and also very, very similar clonotypes grouped together in a single cluster. Now, if we run ImmuneWatch DETECT on this, you can see that 1 cluster will pop up here that we identified using our tool. And these are clonotypes that map to MART1, with the light red ones showing a medium score, and the dark red ones show a very, very high annotation score. So this expanded cluster of 785 TCRs, we believe that it's very likely to be MART1 specific, and actually takes around 10% of the whole T-cell product. And this is now just 1 cluster that we were able to annotate on this public data set. But our collaborators have already told us that they were able to annotate multiple clusters, including viral clusters that could correlate with a very bad product quality. Okay, that's all I wanted to say today. So in conclusion, Miranda has touched upon different methodologies that could be used to define the antigen-specific repertoire, especially in the wet lab. And then I gave an overview of computational methodologies that can be applied here, and brought several use cases where these computational methods were applied effectively. Thank you so much for your attention, and I look forward to engage with you over the questions. Thank you.
All right. So our first question is, how many TCR antigen pairs have been matched based on the most stringent criteria at this point? And is there an available list of those matched pairs?
I think that's a question for me. At this point, does that mean just in general? I've shown you the side of our database. I think we're one of the most extensive database. And we have around 200,000 or more than 200,000 TCR clone types that match to a specific epitope in general. So I think that's the size of the databases now. There's some people, including ourselves, that are working very hard to increase the size of these databases, but that's what we currently have around in the space. There are some other data sets that are known to be much larger, but also very bad quality. So we exclude these data sets because they're famous to be having a lot of false positives in there.
And are these matches HLA filtered, or do any of the prediction programs add in HLA filtering?
That's also an interesting question. So we also actually curate the HLA context on which these epitopes are being presented on, but some epitopes also are presented across different HLA types. So we try to capture this information as well. And we've also recently used that information in a recent new version of ImmuneWatch DETECT where we added a functionality where you can also try and predict what the HLA specificity of your clonotype would be. So we also have an extra column next to the epitope specificity. We will also try and predict what the HLA specificity of a certain T-cell clonotype is. But the databases are also growing in that regard.
So very related question. On average, then, how many TCR sequences are recognized by one epitope? Or let me rephrase that. One epitope is recognized by how many TCR sequences within a specific HLA type?
So I usually look at this from a performance perspective. And so there are a couple of epitopes that have like a huge number of clonotypes defined to that specific epitope. Of course, you can think about SARS-CoV-2 because there's just a lot of studies that focus on the spike antigen and the epitopes derived from the spike antigen. So these are thousands specifically to that epitope. But we also have a few that just have one or two TCRs or maybe 50 TCRs that are specific to a certain epitope presented in a certain HLA context. And I usually look at this from a performance perspective because I usually say that a model like ImmuneWatch DETECT will start to see good performance as soon as you have around 10 to 30 TCRs known to match a certain epitope. We also have a page on our documentation pages, so I'll send the link in the chat, but there you can actually, you have a table that shows what our T-cell coverage is for a certain epitope. And if it's low, we usually found only 1 or 2 TCRs known to bind this epitope. But if it's high, we have more than 100 TCRs known to bind that specific epitope, and that makes it a very, very good epitope to be recognized in your T-cell repertoires, if that makes sense.
So another question about DETECT. How does DETECT decide on or handle similarity between TCR sequences? That is, can it distinguish a clonal family or does it treat each of them as unique?
So ImmuneWatch DETECT is really based on this concept of similar TCRs will bind similar epitopes with some caveat to it. If you find some TCRs that are known to be very, very cross-reactive, we usually give it very low scores. But the first step is usually clustering. So we do some kind of CDR3 and V gene related clustering. So yes, there is some kind of grouping, constructing clonotype clusters that we use to actually infer whether a new TCR would also be able to bind the same epitope.
And that clustering, it's based on clonotype sequence similarity?
Yeah, clonotype sequence similarity, so CDR3 similarity, but also the V gene and the J gene are used for that.
Gotcha. One of our audience members was curious about whether ImmuneWatch also competed in IMREP 25.
Yes, I think I showed a slide on this. So we were also ranked in there. We competed in it. I think we can also send you the link of the competition because it's on Kaggle, so you can actually figure out what the public scores are. We competed in it and with also some structural models there. But we were not, I think we ended up in the top 10 somewhere, depending on how you look at this, because there's multiple submissions from different teams. But we competed in it and we're also waiting for the paper to show the final results there. But I want to stress this IMREP 25 is only on the unseen epitope predictions and not on the seen epitope predictions. And the context that we like to apply the algorithm now is in the seen epitope predictions, because the unseen epitope predictions, as I said before, we truly believe that this is an unsolved problem. And I think this is also going to be the conclusion of the paper that will come out hopefully next year.
Your audience keeps getting ahead of you with the follow-up questions. So on the note of the unseen epitopes, what are the most promising approaches for matching those unseen epitopes? And what percentage of the TCR database does not have an assigned epitope?
Interesting. Let me first answer the last question because our database, yeah, we only collect T cell epitope matches, so it's 100%. All of the TCRs in our database have a match with an epitope. The first part of the question is about what are the most promising techniques, I guess, for unseen epitopes.
Right.
So the conclusion of IMREP25 is that sequence-based models like ImmuneWatch DETECT and other just sequence clustering models show some performance, but not super good. And that the most promising route now is these structural models. Although I would also say that the performance is also pretty bad. So it's not better than, it's slightly better than just a random coin flip. But these structural models, they seem to be the best performing for now on the current data sets that we have. So I would say that structural models like really AlphaFold-like or Boltz or Chai-1, where you try to recreate a 3D structure of your T cell receptor in combination with an HLA allele and an epitope. So it's three different parts that you have to combine. So you need alpha chain, beta chain, you need a full T cell receptor sequence. You construct a model from that together with epitope sequence and then the HLA molecule. And that these models seem to be the best performing, but also very computationally expensive. So we ran some of these models as well. And like I mentioned, our benchmarks show around half an hour to calculate just for one TCR epitope binding. So if you want to screen one TCR for multiple epitopes, it will take a while, especially if you have to look at a whole repertoire. So they're very promising. But I think seen and unseen epitope models now currently have different kind of applications. I think these kind of structural models are more useful in the context of TCR engineering and in T cell therapies.
For the cancer vaccine study, what is the proportion of antigen-specific TCRs observed in tumor and blood and their correlation with patient response?
I don't know the numbers by heart, but it's not always. Yeah, no, I can't answer this. The use case, we have a poster on this, so maybe I should refer to the poster. It's been a while since I had looked at it, but usually what we see is there is some overlap. So we do find TCRs that can be found in tumor. We can still track them in the PBMCs of patients. But what we usually see is that these PBMC clonotypes are not the most frequent one. So if you have a look at a cancer response or a cancer vaccine response, the clonotypes that seem to be vaccine specific are not the most abundant ones either. So they are usually more to be found in the periphery of a PBMC sample. So it's not that you have to just look at the top 20 or top 50 clones of your PBMC samples and hope that these are the ones that can magically eradicate your tumor. No, what we found is that we usually have to dig deeper into less frequent TCRs, clone types.
Gotcha. Are there similar approaches being attempted for B cell receptor clone epitopes?
Yes, I hope so. But we are not doing that because if you have to think about the whole problem of using a computer to infer specificity, if you think about T cells, this is a very defined space because you have the T cell receptor that can only bind to something that's presented on an HLA molecule. So that gives that actually reduces the search space a bit more. But if you think about B cell receptors, these are antibody-like or antibody receptors. This can bind everything like from 3D structures. So you also need to take the 3D structure into account of your target. And this significantly increases the search space. So I've not been following the BCR field for a long, it's been a while, but I'm sure there are some attempts on trying to do that. But at ImmuneWatch, we specifically want to be the best in a certain niche, and that's T cell receptor specificity annotations, because we believe that the problem that you have in our space is we need more data. This is definitely going to be the case in the BCR field as well. I think you need a much more larger data sets to be able to solve that problem. So yeah, I think there are people that tried to tackle this problem, but we're not one of them.
So there's a lot of interest about HLA specificity from the audience. We've already talked about this a little bit, but do your algorithms identify promiscuous epitopes that are recognized by multiple HLA subtypes, such as HLA supertypes?
In theory, yes, because our algorithm is in fact a little bit HLA agnostic, because we've seen that the same epitope can be presented on on different HLA molecules, but that you still have similar TCRs that bind to that epitope. So in theory, our algorithm is HLA agnostic because we just look at the T cell receptor sequence. So yeah, it should go across HLA supertypes, I guess, but I don't have a lot of data to back that statement up.
All right, this next question is a bit long, so I'm going to read it out verbatim so we can work through it together. Do you have any prediction validation data? I'd like to know how sensitive and specific predictions are, especially for targets with less coverage. For example, you showed how COVID predicted clones cumulatively increased in frequency with vaccination. Is that true for all COVID predicted TCRs, or were there a few strong hits among a lot of non-specific or non-induced clones?
Yeah, interesting. For benchmarking, I really would advise you to have a look at this ImRep paper, because that's really the paper where these benchmarks are designed to figure out the data. They're basically reported in AECs, and you can have a look at the epitope-specific AECs. So how good do we score on certain epitopes? These are data that you can find in the supplementary materials. So for really in-depth benchmarking, I would go to that specific paper, IMREP 23 benchmark paper. There is a lot of difference in performance based on how many TCRs you have available for an epitope. And we actually track this as well internally. But for example, in the second use case of the MART1 as well, we are talking with the people that had this data published, and there's some of our most frequent clones. The clones that we predicted to be highly specific, they also had some data backing up in the wet lab where they really cloned this TCR and found it to really match did cloning experiments where they cloned the TCR and tried to do peptide matching in vitro and found these to be confirmative of MART1 binding. I think the take-home message is there is some performance difference based on the number of TCRs that we have available for a specific epitope. And you can find these numbers as well on our documentation pages, hoping to give you an idea on how well we perform for a certain epitope, if that answers the question.
If it doesn't, then we can certainly follow up with more conversation. This is a very engaging Q&A. Let's talk for a second about future state. How do you see these models evolving in the coming years, Sander?
I hope to get more data. I think this is a data issue and not a machine learning problem. So one of our focuses as well is to try to generate more data. I hope to increase. So in the worst case scenario, we just increase the coverage of a tool like ImmuneWatch DETECT, where these seen epitope models, so where you basically get more TCRs known to bind certain epitopes. For example, we are engaged in a few EU Horizon projects. One of them is on malaria vaccination campaigns, where we try to generate the data to be able to track T-cell responses for Plasmodium falciparum. So more data for specific pathogens, more data for specific cancer types. So I hope to see an expansion in data set sizes. What I also hope is that as soon as we reach a certain threshold of database sizes, that we can start to see unseen epitope models that also perform well in certain use cases. I don't think it's going to be like now we have solved the problem. I think it's going to be more stepwise, where these unseen epitope prediction models will become more relevant for more use cases. So I'm hoping to see more data and improved coverage of a tool like ImmuneWatch DETECT, and thirdly, improved performance of unseen epitope models.
So you talked a little bit already about expanding use cases with more data. Miranda, I'm curious to know from you, what applications do you anticipate this technology stack being applied to in the future?
Wow, that's a tough question. So kind of how I was in the introduction, where this is going to be potential biomarkers for, treatment response. I do think I agree with Sander completely. It's really a data problem where it's an information problem where we really need better understanding of these epitope TCR interactions to fully map the space. And as that data gets generated, these are going to get better and better. So I'm excited to see where it goes, but I don't know exactly. I don't think there's going to be an endpoint like that. Hey, this unsolved problem in immunology is solved. I think we'll get to a point where we feel really confident in the different calls that we're making and that we're tracking the right TCRs. So that's what I'm looking forward to.
So one of the audience members is particularly interested in applications for autoimmune disease, a number of which are T cell mediated. What can you guys tell us about TCRs there?
Okay. We're seeing a more and more interest. Yeah, TCR sequencing is also being more applied into the autoimmune field. One of our co-founders is also an MD specialized in autoimmune diseases. The problem there in autoimmune diseases is that we usually don't know what the antigen is that these immune systems are, these T cells are targeting. So that makes it more of a discovery problem because we just, yeah, it's a complete black box. So it's hard for us to generate data on these specific autoimmune diseases. That doesn't mean that we don't have some antigens related to autoimmune diseases in our database. We do have some of them already in there, the known ones. But I think that we need a better performing unseen epitope model to be able to really solve these problems, or big screening panels of different epitopes and antigens. So there's still a lot of improvement to be done there. Although I think there are technologies where you can do like reverse epitope mapping, where you basically use T cell sequencing to figure out what chronotypes are really, really correlating with my disease response or treatment response, and then try to get these chronotypes out there, clone the TCRs, and then try to do the work of recognizing them. So there's a lot of job to be done there, but a tool like ImmuneWatch DETECT out of the box doesn't solve your problem yet.
Yeah, and just to add on to that, I totally agree. It's a really difficult problem because basically you don't know what the epitope that you're going after is. You might be able to narrow that down. So I do see a place for future even wet lab methods that help narrow that space, or basically take large libraries of like expressed protein interacting with TCRs and basically pull those out and sequence those so that you can massively solve this problem, or at least get some idea of protein TCR interactions on a large scale. So I think there will be some advances in that respect that will also help solve some of these problems, because you could take like a proteomic approach where you have a proteome that's expressed of like all surface proteins or small fragments of all surface proteins that the immune system can see already studied on cells. And you could imagine basically if you can interrogate that in a large approach model, like you could generate the data that would be necessary to feed into these other computational models.
Yeah, I want to briefly expand on that, because indeed one of the use cases that we have in motive mode, I think Miranda mentioned it, is the outer circle of the circles that we showed. If you have nailed down a list of potential candidates, motive mode could be used to try and screen for which is now a likely match with some of my TCR chronotypes. So it's a bit more, I would say, experimental, but if you really want to push the frontier further, I think these kind of tools can be helpful. But if you ask for validation data there, I won't have any validation data in that regard. But yeah, if it's a discovery phase, I think it might still be valuable to throw everything you got to the problem.
All right, one last question we have time for. Do either of you have experience using TCR-seq information to inform targeted cancer therapies, such as KRAS, EGFR related drugs?
So we've been working pretty hard on getting more TCRs that bind to KRAS mutated epitopes. So in theory, you could use a tool like ours to just have a look at T-cell repertoires pre and post therapy and see whether any of these KRAS specific clonotypes will get upregulated after therapy. But how this would inform your therapy design, I should think about this further in maybe a private call. I don't know. This is I don't think I have a fulfilling answer for that yet. I have to think about that.
We have a lot of unanswered questions. So again, we will share those with Sander and Miranda so that they can follow up with you. In the last minute, I want to give you both a chance. We have conference season right around the corner. Any activities at ASHG or SITC that you'd like to share with our audience?
Well, we'll be at SITC this year for sure. So I don't think we'll be at the other conference. So I'd be very happy to see everybody. If you're on site, come visit us at our booth. I'll be going to the talks and I'll be around that conference for sure. How about you, Sander?
Unfortunately, I won't be able to attend SITC this year, but we'll be at the World Vaccine Conference in Amsterdam in a couple of weeks. And that's the list that I know at the top of my head. There's probably a longer list at our website. Go check that out, immunewatch.com, if you're interested to meet up, or send me an email, connect on LinkedIn. I'm happy to respond to you. I'm pretty available for responses.
Well, thank you, Sander and Miranda, and thank you all for being the best, most engaged audience and making this a great webinar. I hope you all have a great week. Take care.
Thank you so much. Bye-bye.
Thank you. Bye.